Hi, my name is Jennifer Gray. I am a TEM staff scientist for the Materials Characterization Lab, which is part of the Materials Research Institute here at Penn State. This talk is one I gave for the After Café series, which is part of the Millennium Café series in October of 2023. So what I'm talking about today is how we can use 3D imaging of materials across different length scales using complementary techniques. So we can look at materials at the macro scale and then correlate that region all the way down to the micro level, even all the way down to the atomic level in some instances. So when we're talking about length scales, what we mean is something that's at the bulk level, maybe on the meter scale, millimeter, centimeter scale. So for a hard materials example, here we have a part like a wheel. We go to smaller length scales, we're down to like a test bar or a piece of that wheel. Down to even smaller length scales, we're looking at the grains of the metal that make up that part. Then we're looking at even smaller length scales down in the nanometer range at defects, and then all the way down to the angstrom level to atoms in that structure. So we need techniques that can go across these ranges to see how the microscopic information affects the macroscopic characteristics or properties of the thing we're interested in. On the bio side, when we look at those same types of length scales, we're talking about the bulk length scales, the macroscopic length scales of things we can see with our eyes, like blood vessels. They're getting smaller and smaller, going down, looking at cells, maybe viruses, all the way down to the nanometer length scale of proteins. So how can we correlate all these different length scales to understand what's happening from the micro to the macro? So beyond just looking at these materials at different length scales, it's also important in many cases that we see the 3D structure. And so when we're talking about visualizing the 3D structure of something, we're usually talking about tomography. And so we need tools at these different length scales that can show us the 3D structure through tomography. So at more of the bulk level, on the order of millimeters, that means we're doing something called X-ray tomography, and that's going to help us understand the macroscopic 3D volume and allow us to identify a region of interest that we can then target with other tools that can handle smaller length scales. So we're starting with X-ray CT. We're going to find our region of interest, and then we're going to go to the FIB, or the SEM, which is going to allow us to characterize that same region that we identified with our X-ray tomography at a much smaller length scale, like on the order of microns. So we're going to use the FIB or the SEM to do some serial sectioning, some serial imaging, and we can just look at the region right there in 2D, or we can create another 3D volume using our FIB or our SEM. Again, using serial sectioning to recreate the 3D volume of that precise location. And then if the micron level information that we're getting from the FIB and the SEM isn't enough, we can take it even further, and we can take it all the way down to the TEM, which is going to give us information down at the nanometer level. And often we use the FIB to first create the thin section for the TEM, and then we can do tomography in the TEM to again create a 3D tomogram that gives us that 3D volume information at even a finer length scale. So some of the basics of how X-ray CT works is you have your object, which is your sample, which is in the path of the X-ray beam, which is shining through it, and it's creating the shadow image on a detector. And so you're rotating that sample as you're shining this beam on it and getting the shadow image. So you're getting the shadow image of the X-rays through all different rotation angles. And by having images at all these different angles, we can reconstruct the 3D volume using computer programs. At Penn State, we have 2 different MicroCT instruments for doing X-ray tomography, and you can go to the website here for EESL, which is the Energy and Environmental Sustainability Laboratories, which is part of the Institute of Energy and the Environment at Penn State. And you can find out more about the different tools we have there and the people who can answer any questions you might have. I've listed out here some of the specifications for the 2 different instruments, so you can see kind of what resolution each one might provide in terms of your voxels for your 3D volume reconstruction. So here's an example of something we did with X-ray CT. This was done by Tim Stecco at the EESL. And these are some electrical connector pins that failed. And so by doing the X-ray tomography, what we can determine is pinpointing the location where the failure happened. So this is the part right here, these 6 electrical pins. Once he identified the region that didn't look right, then we can polish back that sample until we expose the region where we think the failure happened at. So once we've identified our region of interest with the X-ray tomography, we've polished our sample back to where we think the failure happened at, then we're going to take it to the focused ion beam. And in this case, we're going to use a dual beam instrument, which has a gallium-based ion beam system in addition to an electron beam, so that we can do both Fib and SEM at the same time. And I have listed here Wes Ocker, who's our technical staff member at MCL, who can answer any questions you might have about this. But on our Fib instruments, we have a resolution of about 3 nanometers. So what we're going to do is we're going to take that polished surface and we're going to mill away some material with the focused ion beam, and then we're going to image it with the SEM in the dual beam instrument. And we can remove multiple layers of materials, milling back until we find the exact region that we think we want to analyze further. Or alternatively, what we can do is we can do serial sectioning in the Fib. So taking multiple slices, multiple images in the SEM, and again, reconstructing another 3D volume only at much finer length scales, in this case on the order of maybe 10 microns or so. So here's a comparison of what we may see. So on the left, we see the X-ray CT image from our sample of those electrical pins. And superimposed upon that, we see the little SEM image that we took by first, again, manually polishing back and then using the Fib to go in and create that smaller section that gives us the higher resolution image that we get in the SEM. So on the right is a zoomed in image of that, where again you see the resolution we get from our SEM image of that location versus the resolution from the X-ray CT. So you see with the SEM we're getting much better resolution, which lets us see in more detail what's going on at that failure site. And then at this location in the SEM, we could also do EDS mapping. So EDS mapping is when we are looking at the X-rays that come off of the sample in the SEM at every point that the beam hits. And those X-rays have a characteristic energy based on what elements they come from. So by measuring the energy of the X-rays that come off the sample, we can create an elemental map that tells us what's going on at those regions. So in this case, you see we've mapped copper, tin, silver, and aluminum. And from this elemental map, it helps us understand better what the problem was at that site that caused the failure. Now, if you have a soft material or some biological type of sample, you may, instead of using the Fib, want to use a microtome. And we can basically do slice and view using a microtome inside an SEM. So this is called serial block face SEM. There's a little cartoon in the upper left corner showing how you take a slice off the top of the sample, you take your SEM image, and then you take another slice. And so you're basically, again, doing slice and view. And you can use that stack of images that you're getting from the SEM to, again, create your 3D volume, similar to what you would do in the fib, except now instead of using an ion beam, you're using a very sharp knife. So again, this is something that would be good for biological types of samples. And again, we have this capability here at Penn State. This is in the Huck Institute. Greg Ning is the contact for that if you have more questions about that. And again, I list some specifications for that instrument below. This is an example that was done. This is a fungus that's attacking ants. And so they did the slice and view, reconstructed the 3D volume, and then you can see how the fungus is located in relationship to the muscle fibers. So if you need even better resolution than what the SEM can give you, then you want to go to the TEM, or the transmission electron microscope. Here you need to have a very thin sample, less than 100 nanometers thick, in order to be able to transmit the beam through it. How do we make that sample? Again, we make that sample in the fib. So with the focused ion beam, we're going to mill out a tiny little slice from the surface of our sample and attach it to a TEM grid. Then we're going to put it in the microscope, and there's a bunch of different information we can get out of the TEM. But again, the types of information we're getting is at the nanometer level or all the way down to the angstrom level. So when that beam goes through the sample, it's going to be scattered by the material, and you can have elastic scattering, which is going to give you a diffraction pattern, and it's going to tell you what crystal structure your sample is. Then the other type of scattering would be from mass scattering or thickness scattering, and so that's going to give different contrast levels based on thicker regions might be dark, or regions that have heavier elements present there would also be darker, so we get mass contrast. The other types of information we can get is from EDS in the TEM, similar to the SEM. We're measuring those energy of those X-rays coming off our sample, which is characteristic of what elements they come from, again giving us that elemental map. Only in the TEM we're going to have much better spatial resolution for that elemental map than we would in the SEM. One other technique we can use in the TEM is EELS. That's where we're measuring the energy of the electrons after they've inelastically scattered going through the sample. That again is going to tell us what elements are present because those energies are characteristic of the elements. And it's also going to tell us bonding information. So that's similar to the type of information you might get from another technique called XPS. So we have a variety of TEMs at Penn State. We have a bunch of different Talos instruments, 2 Talos F200Xs, 1 Talos F200C. They have both STEM and TEM capability with resolutions down to 1.2 angstroms. The Talos C has a retractable cryo box as well and a direct electron detector, so that's good for low-dose imaging, beam-sensitive samples, or biological samples. And then we have our Titan, which is our double aberration-corrected monochromated system. Also has a direct electron detector for doing 4D STEM. We can do EELS and EDS, and because of the aberration correctors, we can get sub-angstrom resolution with our Titan. So here's an example of going from, again, X-ray CT to identifying the region of interest using the fib and then going to the TEM. So this is a graphite sample with some porous regions in it. So we're using the X-ray tomography to figure out where the region is that we're interested in. You can see the picture on the right shows the block. So here we're showing how the resolution difference is between the TEM and the X-ray CT. So on the left we have the TEM image of a graphite sample, and you see the scale bar down there is 500 nanometers. So that same region, if we just got the image from the X-ray CT, you can see the difference in resolution. You really couldn't tell anything about it because it looks so pixely at the resolution of the X-ray CT. So TEM is giving us information all the way down to the nanoscale level that we cannot access with the MicroCT. So with the TEM, we can also do high resolution imaging where we can see lattice fringes of our graphite nanostructures showing the atomic arrangements of the sheets of carbon in that structure. So again, in this image, our pixel size is less than 1 angstrom. So we're operating at that maximum resolution of our microscope now to show the atomic structure of the nanoscale graphite features. And this is an example I took from the Thermo Fisher website. So you see the link down below. So this is not work we did here, but I think it's a really nice example that pulls all these different techniques together. So this is an Inconel alloy produced through additive manufacturing. On the left-hand side, you see the bulk MicroCT image of the part where, again, they've identified the internal region where the structure had some sort of failure. You can then take it to the fib, cut out that area with the fib to expose the region and take an SEM image of it to learn more about the microstructure of the sample there. Or you can cut out a very thin slice for TEM mounted on a TEM grid, and you can characterize it down to even the atomic level, figuring out what phases are present at that crack, figuring out the crystal structure of the phases. So again, you're getting much more information by being able to combine these three techniques together. That's going to let you understand better what the root cause was of the failure for that part. And again, just like with fib and CT, we can also do tomography itself in the TEM. So when we're doing tomography in the TEM, again, we're tilting the sample plus or minus 70 degrees, taking an image at every degree, and then reconstructing the 3D volume from that stack of images. So on the right is the example of some zinc oxide tetrapod nanostructures. And if you just look at them in the TEM, you see these 3 triangular arms with a bright dot in the center. But if you do the tomography and the tilting and the reconstruction, you see that that bright dot in the center is actually a 4th arm sticking up in the Z direction. So a normal TE image is a 2D projection of a 3D volume. By doing the tomography, we can reconstruct that 3D volume. Another example of tomography using STEM in the TEM is some polymer membranes. So this was done by Enrique Gomez's group. They have these very thin polymer membranes that are nanoporous, and so they wanted to see the structure of the pores. So again, by doing tomography in the TEM, they can reconstruct the pore volume. So TEM tomography is also of interest for the bio community. So often in that case, they're doing cryo-TEM tomography because their samples are generally easily damaged under the beam. So if you use cryo, you can try to minimize that damage as much as possible. So Matt Swilius at Penn State Hershey is somebody who does a lot of this. So this is an example of how he looks at protein structures at the tips of neurons. So he combines fluorescence microscopy with cryo-TEM tomography. So he identifies the region of interest first with the fluorescent microscopy by tagging it, and then putting those samples on a TEM grid and looking at that same area in the TEM under cryo conditions, and he can look at the structure of the bundles of the proteins. And so again, he's getting much more information at atomic level length scales by using TEM to analyze these samples. So hopefully those are a lot of examples I've just showed you of doing tomography at different length scales whether it's at the macro level with the X-rays, or the micro level with the SEM, or down to the nanoscale with the TEM. And what's really cool is when we can combine those different length scales together by identifying our region of interest at a larger length scale, drilling down and getting more microscopic information with either the SEM or the TEM that's going to tell us a lot more about what's going on in that sample. So thank you for your time.